Thank you. So sorry for the technical glitch, but I think it works in this sequence also, because I'm just going to make explicit a point that has implicitly already been made by the two previous speakers, which is that if you are interested in precision psychiatry and are going to find stratification biomarkers, that those have to be relatively complex, and they're also going to be strongly multivariate, right? So this is a slide of a type that many of you have seen, and that kind of, I think, exemplifies the philosophy of what we're talking about here today, namely that categorical psychiatric diagnoses aren't specific enough to specify the biology that we need to stratify into subtypes or biotypes that will then support targeted treatments. That's what these colors are meant to signify. And I'm not going to really dive too deep into the slides, but the intuition is, as I said, that there isn't going to be a pink spot. I don't know who still remembers that. That was a promising schizophrenia chemical test in the 60s in urine that turned out to be related to the fact that patients were drinking more tea than controls at the time. Was developed in the UK. It's not going to be simple as that. We're going to be combining data from multiple sources across diagnoses, and that is a better fit with the biology. That's a relatively simple example, combining clinical and imaging data in a sample of patients with schizophrenia, bipolar disorder, depression, and autism, compared to controls looking at a brain phenotype. In this case, is reward signaling. And you can see categorical differences between patient groups. And this is what the field has been done for many, many decades, and started development programs out of findings like this. But if you characterize the individuals, in this case clinically, this is looking at a range of clinical and cognitive scales combined by factor analysis in a factor corresponding to affective instability, cognitive global function, and social function, then of course these factors are related to the diagnoses. Obviously, the ones most impaired in social functions are the ones with autism, ones most cognitively impaired are the schizophrenic patients, and the ones most effectively unstable are the bipolar patients. But you also see this huge overlap. And then if you go back in those data sets and ask what really drives those differences in brain function, then you can see, despite the fact that this is nicely significant, it's not the categorical diagnosis, it's these factors who drive in specific circuits abnormalities in reward signaling. This is really, we think, and I think it's a nice academic industry convergence, what we need to do to become more precise in targeting treatments. Fortunately, we have a lot of data that input into endeavors such as this. We know a lot about the genome. It's also a couple years old, but the genome is now dense with validated common and rare. This is just looking at the common variant associated with psychiatric phenotypes. These are very easy to combine, for example, with imaging data sets. That's big field now, imaging genomics that does that you can use both to discover variants associated with imaging phenotype and to map them across disorders. This is largely in this phase, is like a couple 10 years old maybe, done using linear multivariate statistics, so looking at the effect of each variant, if you will, in each voxel in the brain. And that is no longer where the field is going. It's already been explicit in the last ambition slide of the previous speaker. This is a field that's really being transformed by artificial intelligence and machine learning, because that is a key technology that can discover complex patterns in complex data sets, and it's therefore going to be transformative, I think, for the kind of precision psychiatry we're talking about in this meeting. This is going to be accelerating progress here very much. Just, some examples from our own work, how this is working. This is a consortium that we are heading, Network Consortium in Germany, looking at translational bioinformatics stratification signatures for mental disorders using a technique called transfer learning. Transfer learning sounds relatively simple, but it's actually a very, very powerful AI-enabled technology that allows you to discover discriminating patterns across a range of data sets for a target that may never be represented in any of these data sets individually. You can sort of Lego yourself together a biomarker, even if you don't really have the study or the data set that would reflect what you're really looking at. It's actually quite cool. This is one example, a simple example using epigenetics, something that obviously many of us are interested because we're interested in the interactions of genes and environment, risk for mental disorders. One thing that drives a lot of epigenetics that you can easily measure is methylation of the genome, which is one mechanism through which the environment regulates gene expression. Methylomics, not a problem, something that you can routinely do. Problem is, however, you can usually only do it in blood. As Hugh Marsner said, many humans are quite reluctant to give us brain tissue if we ask them for it. But we're really, of course, interested in predictive epigenetic or epigenomic signatures in brain. Using transfer learning, you can find those, though, because we have all the individual, if you will, Lego components. This is looking for a predictive score for schizophrenia, as an example. So we have methylomic case control data sets in blood. We have methylomic data sets comparing brain and blood methylomics post-mortem. We have data sets for external validation that, for example, links blood methylome with MRI signatures of brain function. And then using transfer learning, you can discover, and really important, out of study validate these signatures across these modalities. So this is, from this study, you can learn a methylomic stratification biomarker that you can show discriminates between patients and controls, that predicts gene expression post-mortem in a way that is consistent with a neurodevelopmental disorder such as schizophrenia, and that can predict in a new sample brain function in brain areas and circuits relevant for schizophrenia. This is something that you couldn't do using linear statistics in any shape or form. This is really something that's transformative using transfer learning. And this is obviously a very rapidly accelerating field that you need a lot of data for. We're here talking also about European initiatives that we're doing. It's not that easy to get data out of Iceland or from France to Germany. That could be more harmonized than it is. But one thing we've been very interested is in privacy preserving federated machine learning, which essentially means that you leave the data where they are in that country, use machine learning and then aggregate it up using transfer learning across sites. And we've also heard about the importance, also the difficulty of being privacy preserving. That is also something that's really accelerating using, what machine learning, specifically transformer architectures, and what's been called generative large language models, brings to the table with regard of dealing with text. So this is something that I think is going to help us in achieving the scale that we need across health system and across countries. And the other thing that is becoming very, I think, useful and important is that these methodologies allow you to integrate in a principled way all the existing knowledge about the biology that you're talking about, using knowledge graphs, which allow you to essentially trawl. This has been done now, and you can just use it as a federated model, the entire medical literature, both abstracts and publications, all all the published association data and use that knowledge graph, for example, in this case, together with gene co-expression networks to really then annotate basically everything with everything that is in an interaction graph, and therefore drive knowledge generation from multi-omic data sets in a way that really hasn't been thinkable, I think, a couple of years ago. So this is a combination, if you will, of AI dealing with textual knowledge, AI dealing with previous large-scale scientific data sets, and your new discovery data set that is much, much bigger than the sum of its parts. And then one final comment, one final footnote to what also the two previous speakers have already said. It's really important to go beyond the individual and characterizing the environment in which this individual becomes ill, becomes well, in which across the lifespan disorders play out. This is just the example of depression, but this is obviously true of all of the mental disorders that we're interested in. And one, I think in this case, very, very useful technology, not the only one, but one that is really very useful is using ecological momentary assessment. You've already seen the example from the PRISM study in which this was done for social phenotype. Smartphones are very powerful technology base to passively, ideally, but also by asking people, assess and integrate a lot of real-life evidence that are highly relevant for neuropsychiatric disorders. Just a few examples from recent works in my group using this and combining a topic of today multimodally to look at things such as social behavior, social context, movement, aspects of the physical and social environment. This is a very useful technique that I think regulators are also open to bring into trials, and that I think we should leverage more in this format to discover the stratification biomarkers that we need in drug discoveries. And in this case, it is also useful that there's a big push in mobile IT to enable the use of AI, of neural networks. So this is very explicitly what Apple is doing. It's very explicitly what many of the Android platforms are doing. And this allows us to essentially at least forward run these relatively complex KI models on the phone, right? So that is going to enable a new generation of predictive biomarkers, because you can immediately integrate the real life information. You circumvent some of the privacy problems that unfortunately exist in this space, because the kind of data that PRISM, for example, gets, it's not so easy to get off the phone, at least in some countries, because they're highly private. In this way, you can avoid this, and you can use this for forecasting and intervention, which essentially gives you a dynamic biomarker, which is relatively useful. Now, in order to do this, we need to get our act together academically, because these data sets are very large and they require harmonization across sites and harmonization of what we're actually measuring. I don't know, the German map has vanished behind this, but this is just trying to show you a new German initiative that started a year ago, which is the German Center of Mental Health, which is funded by the federal government, essentially to do that, which has now progressed quite a bit in defining minimum data sets that are then acquired across all sites and creating the harmonized IT infrastructure, and especially the link between the healthcare IT and the research IT that you need to get these large data sets together. We've been talking, for example, with Marion, a bunch of other European actors to harmonize this across borders. And this way we will get the key ingredient, really, or one key ingredient that we need for this stratification biomarker approach to succeed, which is high quality, large scale, longitudinal data. With that, let me close. Sorry again for the technology snafu. It's easier apparently to talk about technology than to use them to project some slides, but we did manage in the end. Thank you very much.
Thank you, Andreas. And I'm aware that because we're running a little late, but I have actually permission from the sponsor to go over. But Andreas has to leave immediately. So if there are any immediate question for Andreas, this might be a good time to talk about it now. And there's a hand that's gone up. So you need a microphone so people online can hear you.
Thank you very much for the talk. A quick question on you all talk a lot about data, and the German initiative is very nice. Is there an initiative to, so there's the technicality of it for data, etc. But on the medical perspective, is there a common data model, a minimum data set that is planned, be it in Germany or be it international, where everybody collects the same amount of data, or at least the same items?
Yeah, that's an excellent question. We are kind of done in Germany, in the German network, to define a minimum data set core shell. So obviously you know, because of practicalities, you need to keep this relatively short. So this is a 12-minute assessment that can largely be pushed outside the clinician's store. That is a data set that everyone is committed to be getting, and there are going to be shell data sets outside there that expand on this and make this more explicit. Similar processes are running, for example, for ecological momentary assessments. So it's going to be a core data set for that, and for harmonization of assessments, for example, in functional neuroimaging, structural neuroimaging, bio markers, and so on. But that is obviously more of an optimization problem than something that has an explicit solution, right? But that's going to be the German proposal. We've just finished a validation study with around, I think, 1,500 patients to just show that this is feasible, cut it down even more. It is essentially a subgroup of existing clinical scales, right? But you don't need all of those items if you really want to be brief. And this is going to be published relatively soon. It's, however, not at a stage in which I can show you what's in there because it hasn't been published yet.
And do you think there will be a consensus between other psychiatrists in the world? For example, for oncology, they have developed, an example is MCODE, the minimum common data elements, where they try to harmonize at least the collection of.
Yeah, that's the hope. So, for example, Marion, we've been talking to France about bringing this together. This is just then going to be the German proposal. There's going to be, you know, a discussion. We've talked to the Wellcome Trust, who are interested in doing a similar thing, and I've relatively high hopes that once you start this process, we will probably be able to agree on something that we can do. It's just that, you know, this hasn't been a major push in terms of funding. We just got the funding, and we're going to do it exactly as you say. If we want to be at the level of oncology, we need to get a high quality data set that we can all agree on. Because if we only sample like German healthcare, that's not going to be enough. Everyone does something different in Germany. That's not going to be enough. And also, as I think some of the early AI experiences with clinical data sets have shown, you also need a high quality healthcare service, right? So if, you know, people are filling in forms, but they don't have the time or the resources to really diagnose the patient, the quality isn't great, then the best AI is not going to be able to do it.
Thank you.
Thank you for very interesting talks, all of them. I have one question for I guess both the speakers that spoke about the data, the big data. So I'm Anneli from Olink, and I'm wondering if there are blood samples collected in any of these data sets.
I can answer for us. The answer is yes. There's a German regulatory push, which is called the broad consent, which basically allows every patient who is there for purely clinical care to allow you to use the data for research. And by doing that, by our like federal ethics regulator, we're also allowed to take like 2 vials of blood. This is seen to be like a minimum additional imposition, and those can be used. So this is has mainly been a regulatory problem that has been unlocked this way. And similar things, you may know more about the...
Yeah, I mean, I would say yes and no. Yes, because it gets collected at the hospital level. No, because we have not taken that and incorporated that in the NeuroBlue platform. So, but it's certainly possible, very easy to do that. And that's where, if you saw one of the pictures that I've said, which is not only taking the EHR data, so this is a purely electronic health record data, getting the lab data, getting the lab's genomics data, getting the RPM data, and all of that is possible and is part of the plan. That's what we intend to do. But as of now, just getting the EHR data itself was the very first foundation that we wanted to build together, and that has been the case.
Thank you both. So that's excellent. And a follow-up question to that is, we see a lot from our perspective, the problem of pre-analytical variation. So as you embark on these fantastic and ambitious initiatives, it is our perspective is that it's very important to harmonize the SOPs for those collections. Of course, plasma EDTA collection or similar things is quite standardized, but we often see a signature that is site-specific. So especially as you look broadly at the proteome or many other omics, I assume.
And this was one of the cases that what we are trying to push at the clinical practice is exactly that. That how do we truly standardize the new set of data collection itself, that which even Hugh was mentioning, that if you start at the origin itself, you can enhance the data quality metrics or the parameters by which it gets collected, then all of a sudden the potential outcome that we can have or the AI models that we can build is going to be so much more sophisticated than what is possible right now.
Yeah, and even if you have that, it's important to interface it with the clinical processes in a way that actually works, right? So we are committed to do this in everyone who comes through the door. We have about 100,000 patients who come through the door every year in the hospital that I lead. And so we've just actually published how you can, as a process, put this into the normal clinical admissions and diagnostic flow, so that that isn't impeded and it doesn't, crash your main income stream, right? This also, but it is possible in principle, but it isn't just, okay, hey, take 2 vials of blood and put them in a fridge and send them to me sometime, as I think you're also pointing out, right?
Yes, so that is fantastic. I think that's really an essential step. So I would love to follow up on that. Thank you.
Thank you. We're a little over, but let's get going. And thank you, Andrea. Safe trip home.
Thank you.
Hope your flight goes on time. There's a bit of a deluge warning in Germany. So if you live there, make sure the windows are closed.